The Center for Integrative Environmental Health Science and the Department of Pharmacology and Toxicology welcomes you to our Environmental Health Science Series seminar. It's my pleasure to introduce our speaker, Dr. Carolyn Klinge. Dr. Klinge is a university scholar and a professor of biochemistry and molecular biology here at Louisville. She also currently serves as a leader of the CIEHS Cancer Research Interest Group and is a member of the Brown Center Experimental Therapeutics and Diagnostics Program. She received her Ph.D. from the Pennsylvania State University and then headed back to, went to undergraduate in New York, but then she headed back to New York to do her postdoctoral training at the Department of Biochemistry and the Cancer Center at the University of Rochester School of Medicine and Dentistry, where she also held several academic positions following her postdoc work. She came here to UL to be with us here at UL in 1996 and has climbed the ranks to full professor about 2005. Her research interests include molecular mechanisms of estrogen action, investigating mechanisms of resistance to endocrine therapies, and targeting estrogen receptors in breast cancer and estrogens in lung adenocarcinoma. And she's also interested in sex differences in liver responses to environmental pollutants. Throughout her impressive and productive academic career, she's published over 140 reviewed scientific articles and reviews and has contributed to several book chapters. She has a lengthy list of NIH and DoD grant panel appointments and has served as an editorial board member and reviewer for several different scientific journals, and currently serves as the co-editor-in-chief for the Molecular and Cellular Endocrinology. Her research has been largely funded by several grant awarding agencies, so NIH, DoD, Susan G. Komen Breast Cancer Foundation, and various things, just to name a few. And she currently has several grant funded research projects ongoing at this time, including the Summer Endocrine Cancer, including the Summer Endocrine Research Scholar Program and a DoD Breast Cancer Research Program grant that addresses the role of M6A in the transcriptome of endocrine sensitive versus endocrine resistant breast cancer cells. And today, she will share her research from yet another current ongoing NIH funded research program that examines how diet and exposures to polychlorinated biphenyls regulate liver Epitranscriptome in mouse models of non-alcoholic fatty liver disease. So without further ado.
Thank you very much for that extensive introduction. When you get to be as old as I am, you have a lot of data and a life under your belt. So I'm very pleased to be able to share some of the research that I've done, that it was funded by an R21 that Matt Cave and I were fortunate enough to be awarded from the CIEHS. And this work was also supported by the CIEHS P30 Center from 3 medium vouchers. So I have nothing to disclose. Now for this audience, Dr. Cave has already presented a number of times about the prevalence of non-alcoholic fatty liver disease and its a growing preponderance in the United States. So globally, about 25% of people have NAFTA, and in the United States, about 24. Now, non-alcoholic fatty liver disease encompasses fatty liver, non-alcoholic fatty liver, where the liver has steatosis, which is just fat accumulation in the liver. And that can progress in about 25% of cases to NASH, non-alcoholic steatohepatitis, where there's an increase in inflammation, hepatocyte injury called ballooning, and this can involve inflammatory cell infiltration as well as fibrosis. More fibrosis gets into cirrhosis, and in some cases, about 3% of cirrhosis can advance, or NASH can advance into hepatocellular carcinoma. Now, non-alcoholic fatty liver disease is a metabolic disease, so there are a wide variety of accompanying metabolic diseases, including type 2 diabetes and cardiovascular disease. The prevalence of NAFLD is higher in males compared to females, although in postmenopausal women, that incidence of NAFLD starts to rise and comes up to that which is seen in men. Now, what Dr. Cave's group has published is that hepatocellular signaling is inhibited by metabolic and endocrine signals and exposure to PCBs, resulting in toxicant-associated steatohepatitis, or TASH, which is another form of NASH associated with toxicant exposure. And this is, he has published a number of publications in PubMed on this topic using mouse models, but in addition, a work that he's done with the Aniston Community Health Survey has shown a significant association between 35 orthosubstituted PCB congeners in humans and liver disease in that population, namely NAFLD and NASH. So what are polychlorinated biphenyls and why do we care about them? Well, here's the structure of a polychlorinated biphenyl. So we see two phenolic rings and we see a number of places where chlorine can be bound. And so in fact, depending on the position on the ring, there are 209 congeners of PCBs. They come in two basic categories. The dioxin-like or planar PCBs, the prototype is PCB 126, which you'll see used in the experiments that we did with Matt Cave's group, or the and the dioxin-like PCBs activate the aryl hydrocarbon receptor, one of pharmacologists' favorite nuclear receptor, although it's not a nuclear receptor of the steroid receptor superfamily, as is my other favorite receptor, the estrogen receptor. But we also have non-dioxin-like PCBs, and these are non-planar. And the prototype is the PCB mixture Aerochlor 1260. That contains 99% non-coplanar congeners, and it mimics this mixture of PCBs, mimics the PCB bioaccumulation that has been detected in human adipose tissue. Now, polychlorinated biphenyls are POPs, they're persistent organic pollutants, so they will accumulate in adipose tissue in animals and in humans as well. The non-dioxin-like PCBs activate the constitutive androstenedione receptor and PXR and inhibit epidermal growth factor signaling. And that's work that was done by Josiah Hardesty in Matt Cave's lab. PCBs not only are metabolism disrupting chemicals, as I mentioned earlier in this exposure model of toxicant-associated steatohepatitis, but they're also endocrine disrupting chemicals. So they have impacts on endocrine signals, both in females in reproductive tract and in males in reproductive tract. Now, where do PCBs come from? Well, they were manufactured in the United States, and their manufacturer was banned in 1979. And I certainly remember this because I was in college, and we heard about the accumulation of PCBs in sediments of the Great Lakes and in the sediment of the Hudson River Valley, where electrical equipment was manufactured by General Electric, like this transformer here on a power line. And it polluted, it was down in the sediment of the Hudson River, and it required great effort to clean it up, and I'm not sure it's all cleaned up. So these industrial chemicals, these PCBs, are contaminants in food. They're POPs, so they are persistent. They are fat soluble. This is certainly not aqueous soluble. And so they accumulate in fat, in meat, in dairy, and fish, and we intake those foods into our bodies. And so it's associated with steatohepatitis in name. Dr. Cave's group has used an environmental model of PCB exposure in the context of high-fat diet in mice to show the progression of non-alcoholic fatty liver disease into steatohepatitis. And this is accompanied by changes in the gut microbiome. This is work from Dr. Banrita Walling, first associated with Dr. Cave's lab, and now on her own as an assistant professor and continuing to collaborate with Dr. Cave and his group. And I came along and I thought, I wonder if there is a change in the epitranscriptome and in the microRNA that are going to influence the expression of proteins in this model of non-alcoholic fatty liver disease progression to NASH. So I hypothesized that PCB exposures in the context of high-fat diet modifies the RNA epitranscriptome, resulting in altered transcript and protein abundance associated with biological processes, stimulating NAFLD progression to NASH or TASH. And so fortunately enough, the NIH reviewers gave us a good score, and we were funded with an R21 grant. So what is the epitranscriptome? So Transcriptome. So epi means over the transcriptome. So these are chemical modifications of the 4 bases in RNA. So we see adenosine, guanine, cytidine and uridine. And we see many methylations here on these data. We see some other modifications as well here. So the uridine, you'll see that modification. In humans, there are over 170 chemical modifications of transcribed RNA. And this is all transcribed RNA. So we know in the cell, most of the RNA in the cell is tRNA and RNA. And mRNA is less than 5% of the total mRNA or total RNA in the cell. And then we also have non-coding RNAs, and the list of non-coding RNAs includes link RNAs, microRNAs, circRNAs, and a whole host of others. But what you can see here is that most of the chemical modifications occur on tRNA, and then rRNA, and then thirdly, mRNA. So when we see global changes in the epitranscriptome, that's all the RNAs. It's going to be primarily, if we look at it, tRNAs and rRNAs and a little bit of mRNAs. We have to dissect out what's going on. So tRNAs are the most heavily chemically modified forms of RNA. And what we can see on the anticodon loop is most of the chemical modifications, or many of the modifications, occur here. And this would be important because that anticodon loop is what binds the RNA, mRNA, to allow the translation of that message into protein. So what these chemical modifications do is control the secondary and tertiary structure of the tRNA for that interaction, and thus for protein synthesis or translation. Oh, and chemical modifications also occur on mitochondrial RNA as well, a topic of interest, but we're not going there today. Now, the most common epitranscriptomic mark is N6 methyladenosine, or M6A. And in mRNA, about 0.1 to 0.4% of the A's are modified to M6A, and that gives us about 3 to 5 M6A sites per transcript. But M6A is found on all RNAs as a modification found on RNAs. So tRNAs, rRNAs, non-coding RNAs, and mRNAs. Now, M6A has become one of those interesting hot topics. If we look in PubMed, we can see by years of publication that it's really accelerated. 2015 is when I first became aware of M6A modification, and it's important for regulation of processing of primary microRNAs into precursor microRNAs within the cell nucleus. And if we look at the number of publications on M6A in human liver, well, you can see it's going up. And what about M6A in NAFTA? Well, it's also going up. We published our first paper in 2021. So we're probably including these little dots that you see here. So the first question we asked under that R21 grant was, does high-fat diet and PCB exposure alter the liver epitranscriptome? And so we came along and joined with the work that had already been accomplished in Matt Kay's lab by Jin Zhang, Ben Rita Wallen, and Kim Head. And what they had done was to take 8-week-old C57 black 6 male mice and put them on a high-fat diet, so 42% fat. And after 2 weeks on this diet, the mice received a single oral gavage of either the vehicle control corn oil, or Airchlor 1260, or PCB 126, or the combination of Airchlor 1260 plus PCB. And the concentrations that were used to treat the mice, or the doses that were used to treat the mice, are those that allow the mice to mimic the exposure levels that are seen in humans. After 10 weeks after that oral ingestion of the PCBs, so a total of 12 weeks on the high-fat diet, and they were kept on the high-fat diet. The mice were fasted for 6 hours prior to being euthanized, and tissues were then collected. At the time my group joined this project, Matt Cade's group had already done a protein extraction, that was Jin Zhang's Ph.D. dissertation, and to identify the proteome of the calf livers in this mouse model, and that was work with Mike Merchant. We came along, and Kelly Peel isolated RNA. She isolated total RNA either for polyA selection and mRNA transcriptome identification, or what I'm going to talk about right now is our total RNA isolation for LC-MS analysis to identify global RNA modifications against control known modification standards by the Center for Regulatory and Environmental Analytic Metabolomics, which is headed by Dr. Zhang Zhang. And Klein, he actually performed the assay in Dr. Zhang's lab in the cream core over on the Belknap campus. And this was supported by a CIEHS medium voucher. So what did we find? Well, we identified 10 RNA modifications in the total liver RNA. So remember, this is all the RNA. So this is RNA, tRNA, mRNA, non-coding RNAs. We don't know which of these kinds of RNA are modified, but these are the chemical modifications that are detected on the total RNA. And what you can see is in yellow, we have the Nrf2 1260 exposed livers. And all these values are compared to the high-fat diet vehicle control, so the quartile control ones. What we can see is that, and the color, so red is up and green is down. And what we can see is that we see two modifications in all three PCB exposure samples, and that's AN and M6A. Well, I've already told you about M6A, so you know all about that. But what's AN? Well, that's 2-O-methyl A, and that's found in tRNA and small nuclear RNAs. And what we can see is that AN was increased both in the Nrf2 1260 and the Nrf2 1260 plus PCB 126 exposed livers. But in the PCB 126 exposed livers, we found the most RNA modifications, and they were all reduced in prevalence, in abundance. And with M6A, we see a decrease in the Nrf2 1260 exposed mice and the PCB 126 exposed mice, but the combination did something different. We see an increase. Now, some of you think, well, these are really teeny tiny little numbers, but remember, we only have 2 to 6 M6As per mRNA. So really, and I know this is not just mRNA, but these chemical modifications are rare. So small numbers are actually quite significant when you think about it. Oh, and the analysis was performed by Dr. Shesh Rai and John Min Penn in the Biostatistics and Informatics Facility Core for CIEHS. Now, thinking about how M6A happens and what does M6A mean, we want to look at M6A epitranscriptomics, and that involves writers, readers, and erasers. So the writer complex is the RNA methyltransferase complex that transfers the methyl group from SAM, the universal methyl donor, to the adenosine to form M6A within a specific sequence in the mRNA called the RAPT sequence. So that A is going to be the target. Now, the M6A mark is dynamic. So it is put on by the metal 3 complex, and it is erased by 2 eraser proteins, ALK-BH5 and FTO. And for those of you who are really aficionados of M6A, we know that FTO may or may not be really the M6A eraser that people initially thought it was, because it really seems to erase M6AA, but which is at the 5' cap of all mRNAs. But that's controversial, so they're still both listed as erasers. What's really important here is readers. There are 18 known readers of M6A. What these are RNA binding proteins that recognize that M6A, and they regulate then what happens to that transcript. They interact with the splicing machinery to allow splicing to happen or to inhibit splicing from happening. They're involved in export of that mRNA. And then we have cytoplasmic readers that either can stabilize or destabilize that transcript to stimulate or inhibit translation. And you may notice that some of these readers that are listed here in the little yellow boxes are the same. And that's because how the reader interacts with that mRNA in the subsequent pathway that messenger RNA takes is dependent on the cellular context, other interacting RNA binding proteins, and other interacting proteins. So it's really complex, and it has to be looked at much like microRNA and a case by case basis. So transcript by transcript basis and cell type context specificity. Well, then we wanted to integrate what we had found in the epitranscriptomics with what we had seen from mRNA-seq and the protein abundance that Zhen had published from Matt Cave's lab already in the proteome analysis. So we looked at the expression of writers and erasers and readers for each of the differentially abundant marks that we had found in the total epitranscriptome. And the bottom line here, some things agree, some things disagree, many things disagree, but the only one that really agreed across the board was down here, FMR1, which is a reader of M6A. And we can see that its transcript abundance was increased in Aerochlor 1260 or Aerochlor 1260 plus PCB exposed mice. And likewise, its protein abundance was increased. So conclusions from this first study was that we were the first study to identify global RNA epitranscriptomic changes in the livers of mice exposed to high-fat diet and PCBs that cause CASH. And we found, we identified.
I think you were the first to show liver epitranscriptomic changes from any environmental pollutant.
That's true. So we're the first group to show any environmental pollutant change, the epitranscriptome in liver. So we identified 10 of altered RNA changes in the liver. We detected that the abundance of M6A and AN and M6A were different between the PCB exposure groups, and those were the only consistent marks that changed across all PCB exposure. And we found that increased M6A in the co-exposed PCB 126 and Aerochlor 1260 livers was associated with increased transcript and protein abundance of the reader FMR1. Now, the big limitation of this analysis is that we don't know the location of these post-transcriptional marks in the global RNA. Are they on tRNAs, rRNAs, mRNAs? Get into that later as we go on. So then we ask the question, well, what's the relationship between protein and mRNA expression? When we think about it in our minds, we envision that mRNA is going to be translated into protein. And so we sort of expect some kind of linear relationship between mRNA abundance and protein abundance. Well, I'm here to say it's not true, and it's not true in many, many, many studies. You really have to look at your transcript and protein separately. Okay. So Belinda Petrie, who is writing her dissertation now and is eager to get on to her postdoc in Eric Rushka's lab in bioinformatics, integrated mRNA and protein interaction, did a protein integration analysis to look at differentially expressed genes and differentially expressed proteins with the 3 different PCB exposure groups. So these are the high-fat diet mice. So this is all versus control. And we have the Aerochlor PCB 126 or Aerochlor 1260 plus PCB exposure groups. So we see only one gene and protein in relationship, namely an increase in FMR1, that M6A reader in the Aerochlor 1260 group. We see 7 transcripts and proteins in the PCB 126 group, and we detected 56 transcripts and proteins in the same direction in the Aerochlor 1260 and PCB. So the most genes and proteins in the Aerochlor 1260 group. Every time we look at the Aerochlor 1260 and PCB co-exposure group, we see the most changes. mRNA, tRNAs. We just did that with Dr. Julia Charkar, who's here today. So in other words, this integrated mRNA transcriptome and proteome analysis shows low concordance. So we need to examine what's going on between transcript abundance and protein abundance. So one thing that we know regulates transcript to protein abundance is microRNAs. So Belinda also did an integrated microRNA protein interaction analysis and identified microRNAs that were putatively targeting their microRNA response elements in the 3' UTR differentially expressed proteins based on that study that John did in Matt Cave's lab, and that was published, the proteome study. So If a microRNA and proteins are in opposite directions, so if a microRNA is decreased, the target protein should go up. And if the microRNA is increased, there's going to be more to bind to the transcript to block translation, so the protein will go down. And so you see very low concordance again between identified microRNAs in our sample exposure to our aircore 1260 and increased protein expression with decreased microRNA 13 relationships. PCB 126, increased protein, decreased microRNA 4, decreased protein, increased microRNA 13. In the aircore 1260, 378, where we see an increase in protein and a decreased microRNA. These are all putative because these are changes that we see in the microRNA, changes that we see in the protein, but we don't know if they're really targeting. There are databases that you use to identify interactions, but unless you experimentally prove those, you don't really know if a particular microRNA targets a particular gene with transcript, in other words, a protein. So what you can see on the lower right-hand side here is that Belinda created in BioRender an alignment between the seed element of a particular microRNA and its putative target in the 3' UTR of genes. And these genes were selected because they had a role in liver and in non-alcoholic fatty liver disease. And we're going to see these genes on the next slide. So what you have to do is to validate a reciprocal protein microRNA relationship by a dual luciferase reporter assay. So the putative microRNA response elements from 4 genes were cloned into what is called the p-mirGlow dual luciferase microRNA target expression vector. What that vector has is a firefly luciferase, where we clone the 3' UTR downstream. And the microRNA, if it works the way we think it will bind to that microRNA response element within the RNA-induced silencing complex, the RISC complex, and we'll see a decrease in luciferase activity. And so we selected 4, 3 where we detected an increase in protein expression in the airflow plus PCB 126 exposed livers, NAL3, SMARCA4, and NOX3, and 1 where we see a downregulation of STM1, and its target was MIR9. And so if these are bona fide targets or validated targets, we'll see a decrease in firefly luciferase activity when the specific microRNA is co-transfected into the HEC293 cells with the reporter assay versus a control, a microRNA control. And how this dual assay works is not only do we have this firefly luciferase, but the same vector contains ranilla luciferase that's driven by a CMD promoter, so that that's always on. So that's going to be your control for how much plasmid you're getting inside the cell. Okay, so we've seen a decrease we've validated for targets. So a conclusion from part 2 of the work that we've done, and this was published in Environmental Toxicology and Pharmacology this year, is that PCB exposures alter liver mRNA and microRNA abundance in pathways that are indicated on this lovely BioRender diagram that Belinda created. These are pathways that are known to be associated with NAFTL and NAN. We found that integrated transcriptomic proteomic analysis showed low concordance. So therefore, transcript abundance is not a good proxy for protein abundance in this model. We really need to look at protein. And we confirmed some reciprocal microRNA protein interactions that were associated with proteins in non-alcoholic fatty liver disease. And we can conclude that additional mechanisms are involved in protein changes in NASH and in the pathways that contribute to the progression from NAFTL to NASH. So the next question was, well, what about M6A? Right? Do high-fat diet and PCB exposure alter M6A in transcripts? In other words, in mRNAs, in pathways in non-alcoholic fatty liver disease. So it's the same model, it's the same liver samples that were initially collected by Dr. Cade's lab. And what Kelly did was to perform RNA isolation, poly A selection, and then M6 RNA immunoprecipitation sequencing. So here's how this works. She took liver, did RNA isolation, got RNA, did poly A selection, fragmented that, collected fragments, and then did the M6A RIP. So what this is RNA immunoprecipitation. We have an antibody that recognizes the M6A. So everywhere there's an M6A, the antibody will bind and will pull down those fragments that have the M6A. And we do this versus IgG, of course. And so then we reverse transcribe. Of course, you get rid of the antibody, and then you reverse transcribe those or into cDNA, and then sequencing by Illumina NextSeq, and then bioinformatics. And the bioinformatic analysis in this case was provided by Klinge and Driva and Eric Rushka in the KYINDRE bioinformatics course. So here's a heat map of the M6A peaks in liver genes identified by this M6A RIP-seq. In that, from the high-fat diet mice control in the first lane, the Aerochlor 1260 exposed samples in the second lane, the PCB 126 in the third lane, and the dual exposure to Aerochlor 1260 plus PCB in that fourth lane. And when I saw this, I was ecstatic because this is, in my knowledge, the first time anybody has ever seen differences in M6A on genes in any kind of experimental model. So this was super exciting because you can see it's just very different. So this definitely means that PCB exposures change M6A deposition and abundance on genes in the liver of these mice. So we can then analyze it in different ways. And when I looked at the literature to figure out, okay, now we have this data, what do we do with it? So, I like to go to other papers of similar nature, although not in liver and not with any sort of toxic exposure, and find out what they did. So what people do is they plot the genes with M6A at different chromosomes. And what we know from this analysis is that most of the M6A peaks were identified in the coding sequence in the 3' UTR, and 97% contained the conserved RACH motif. So that's the site where metal 3 is going to bind and deposit that methyl on the N6 position of adenosine from SAM. And we can also look at the number of genes and the number of M6A peaks. And what we can see is most genes have one M6A, but some have quite a few M6A peaks. And what we can see is that we see more genes with peaks in the PCB 126 and in the PCB plus Aerochlor 1260 exposed, co-exposed liver samples. And then Klinge did this Venn diagram to look at the M6A containing genes and percent of total genes were grouped to look at those that were unique in each exposure group or those that were in common. And then, of course, I wanted to know, well, what pathways are associated with the genes that are altered with M6A peak density? So I ran the data through Metacore, and you can see in the pathways indicated that there are a number of pathways that would be found in non-alcoholic fatty liver disease. So that was exciting. And then Belinda did an integrated analysis of the M6A containing gene and the differentially expressed genes that we obtained from the mRNA-seq, just looking at the Aerochlor 1260 plus PCB 126 co-exposure. And again, the reason for that is we see the most changes in this co-exposure model in the mice. And so down here we have transcript abundance. So here we have more transcript abundance, less transcript abundance. And then the size of the dots, red is more M6A peaks, and less is green. And then what Belinda did was indicate some genes of interest, and FZD1 was interest BLG1. So more M6A peaks, more transcript abundance. But on the other hand, we see over here, BCL9, for example, more M6A peaks in the PCB 126 plus Aerochlor 1260 co-exposed samples versus the high-fat diet control, and yet less. Well, okay, so why is this? Remember that the M6A can either promote deposition, depending on the reader, will either promote degradation or promote translation. So it just all is context dependent. So then I wanted to look at what are the top 10 hypermethylated M6A peaks in liver genes. And so that's summarized here. We have Aerochlor 1260 in the number of peaks, PCB 126, and then the co-exposed samples, Aerochlor 1260 plus PCB 126. What we can see is that we see an increase, or this is all increased number of peaks compared to the high-fat control. What we can see is we detect an increased number of M6A peaks in ApoB, in all three groups. Now, the number of peaks changes, which is interesting of itself. And we know that ApoB transcript expression is decreased in the PCB exposed, PCB 126 exposed sample and in the co-exposed airport plus PCB exposed samples. And then we could also look with the M6A RIP sample, so the input into the M6RNA Illumina RIP-seq samples to see what we found there. And what we can see is that if we look at the enrichment compared to IgG-IP, is we see a decrease in ApoB transcript in the Aerochlor 1260 plus PCB co-exposed samples. So here it looks like M6A, even though we see more M6A, we're seeing a decrease in transcript. So maybe something's binding to that M6A that's causing a degradation of that transcript. And do we see a decrease in protein is the next question. Oh, well, first I want to say, well, why do we care about ApoB? Now, I don't teach the lipids part in the biochemistry in the integrated M1. Dr. Barbara Clark teaches that. So I've certainly been talking with Barbara about why ApoB is important. But I get that it is important. I've heard of it before. What it is there's one gene, ApoB, and it is transcribed in every tissue or many tissues, and it is constitutively transcribed. So therefore, post-transcriptional regulation of the transcript to protein is going to be what's important. So my theory is that M6A is one of those important features for post-transcriptional regulation of ApoB production. But it's differentially, so the whole transcript is expressed in the liver, but in the intestine, what happened is that there's a protein called A1CF, and that stands for ApoB1 complementation factor. It is an RNA binding protein that was going to modify that C to a U, and UAA is a stop codon. So what happens in the intestine is that you form a shorter transcript and make a smaller protein, ApoB48. And ApoB48 is what coats the chylomicron in the intestine to allow triglycerides and tags, triacylglycerols, to be take the fat from our intestine to go to the liver. And then in the liver, we have the full-length ApoB, and that makes the ApoB 100. Now, the ApoB 100 is a very large protein. It coats the VLDL, which comes from the liver with triglycerides and cholesterol esters, and then is converted in the plasma to a bunch of intermediaries. And ApoB is actually what binds to the LDL receptor for internalization into the hepatocyte. And reduced ApoB protein plays a crucial role in hepatic lipid accumulation in NAFLD, because you can't send out those lipids from in the form of VLDL. Okay. So then Belinda did MET-peak analysis to identify the positions of the M6A in rash motifs in the M6A RIP-seq data. And then Kelly performed, and she did the primer design. So we have different primers. The primers are in brown in 3 different regions of the ApoB transcript where there is a rash motif. That is underlined right here in the 3 sets, and designed primers. And so Kelly ran quantitative real-time PCR looking for M6A enrichment compared to input. And what we can see is with primer set A, we see an increase in Aerochlor 1260. So this is just looking for M6A enrichment in these pieces of the mRNA in the samples. Remember, we see an increase in M6A in all 3 groups, even though we're seeing a destabilization of the transcript. Will we see a decrease in protein? We just want to find out if M6A is really there, right? So, and we see increase in signal in this primer set in PCB 126 and primer set C in PCB 126. Now, a caveat of this is that we detected, so we're only analyzing 3 of 10 identified M6A sites, and the other sites may have a greater impact on transcript abundance. So there's never any ending to experiments that you can plan to do. That's what we need to do. So we also observed an increase in the number of M6A peaks in A1CF transcript, and we then looked to see if we would see an increase in A1CF protein expression with PCB exposure, and we did. In the samples where we saw an increased number of peaks from 1 to 2 in the PCB 126 exposed samples, and from 1 to 4 peaks in the airflow plus 1260 samples, we see an increase in pulse. So this is sort of the opposite story of ApoB, right? In ApoB, we see increased M6A, but decreased transcript and potentially decreased protein. And in A1CF, we're seeing increased M6A, but we're seeing increased protein. So maybe different readers or cellular context or interacting factors are regulating this. Well, and I was thinking about this data, and I'm trying to write an R01 now, because the reviewers rejected triage the ESR01 I put in twice to do more of this kind of work. And it's clear to me they want very specific, mechanistic, specific aims. So they want to know what is, one M6A mark on one transcript doing. So that's the kind of thinking I have to be thinking about. And so I was thinking about this, I'm thinking about A1C and its role in processing of the ApoB transcript, inserting that U to make a stop codon to get the ApoB 48. But yet it's in liver, and ApoB 48 is not made in liver, it's making that full length ApoB 100. Well, there was a paper published in 2019 showing that it actually does CU editing on 16, and here's the paper, 16 genes. And so here are the identities of 16 genes that they reported in mouse liver that were regulated by this A1CF CTU RNA editing. So I asked Belinda to go and integrate through our data, and which she did in lickety split. She can do this quite quickly now. And look at whether we see increased transcript and protein levels of hepatic genes that are regulated by A1CF. And the answer is yes, not for all of them, but for some of them. And what about protein? Again, Belinda could integrate with the data from Zhen's paper from Matt Cave's lab. And we see an increase in TM, EMA, and CIP at the protein level. We see an increase in transcript. We probably haven't looked at all these proteins. So this is very interesting because TM38 interacts with a bile salt export pump, and CIP promotes oxidative damage in pre-tumor NASH, leading to HCC. So these could be important, mechanisms which reviewers want in the conversion of NAFLD to NASH in this experimental model of high-fat diet and PCB exposure-induced NASH with fat. Okay, so we showed M6A modifications play a role in PCB reprogramming of hepatic gene expression in this mouse model. We found pathways associated with changes in M6A peaks in all 3 PCB exposure, included those which would be found in NAFLD progression to NASH. We found that PCB exposure alters hepatic M6A epitranscriptome in a non-dioxin-like versus dioxin-like PCB selective manner. So different PCBs, different M6A, different genes. And we showed that M6A RIP-seq qPCR of the region for the ApoB transcript harboring M6A peaks. So that validated our M6A RIP sequencing results. Now, next is a different model. Rather than high-fat diet, we have a low-fat diet model. And this is work done with Kim Head and Rita Walling. And Kim and Belinda and Kelly actually treated the mice. So in this case, they're on a low-fat diet. We started again with 8-week-old C57 black 6 mice. Now on a low-fat diet, they only have 10.2% fat in their diet. And after 2 weeks, they were exposed to a single oral administration of Epitranscriptome 1260. Now, I know these numbers are unequal, and that's okay. We've got 10 in the control and 30 in the exposure group, and that's because we want to get lots of samples, because you need a lot of samples to do a lot of work with. And during the course of this overall 36 weeks on the low-fat diet, and again, Epitranscriptome 1260 at 2 weeks, a number of tests were run by Kim and Belinda and Kelly. And then again, mice were fasted for 6 hours prior to euthanasia, and then livers were collected. And again, we isolated, Kelly isolated total RNA, either for ultimately Illumina mRNA-seq or to be sent to Cream for LC-MS analysis of the global RNA epitranscriptome, again, global RNA transcriptome. And this was supported by a medium voucher from CIEHS. And this time the work flow was done by Rebo Zhu, who is a master's student in Zhang Zhang's lab in Korea. First of all, unlike the previous study where the phenotype of the mice had already been published, this was new. And so Kim did IHC, and the slides were read by Dr. Yan Li to look at steatosis and fibrosis in the samples. She did quantitative real-time PCR of POA1, which is a fibrotic marker, and we see that it's increased in the Aerochlor 1260 exposed samples. We see that an increase in AST in the serum of the mice. We see increased steatosis, increased inflammation, no significant increase in ballooning in the long-term Aerochlor 1260 samples, and an increase in the NAS or NAFLD activity score in the samples. And then Dr. Lee was able to look more closely because we have 30 samples. We could pull out samples that had from the Aerochlor 1260 exposed mice that had low NAS versus high NAS score. And the reason we did this is because it seems like not all the mice have comparable degrees of steatosis. And so if we separated out the NAS score into low and high NAS score, the high NAS score mice had an increase in body weight and they had higher cholesterol in their serum. So there's something going on. We don't know what it is. One of the things we'd like to do is send those liver samples for identification of a PCB in the livers. Did all the mice really have the same amount, or can we correlate, for example, low NAS in the Aerochlor with low PCB congeners in the liver? So of course we have to get a grant to do all this stuff. So of course we ask the question, does the Aerochlor 1260 long-term exposure affect the global Epitranscriptome? And the answer is yes. And what's interesting is the chemical modifications are different. We're picking up different chemical modifications than that which we saw in the first high-fat diet study. Two are the same. AM, we saw that one before, and M6A. What we see is M6A is decreased and AM is decreased. So previously, we saw a decrease in AM in the Aerochlor 1260 exposed samples. So diet is impacting RNA modifications, and also the length, maybe the length of the diet. I've also put down here what type of RNA you would find these modifications in. And again, you see that many are in tRNAs. We also did the mRNA-seq on the analysis on the low-fat diet versus Aerochlor 1260 exposed samples, thanks to Drs. Kalpani Silva and Julia Charkar and Eric Rushka. And so there's the top upregulated and downregulated genes on the left and the right respectively. And this is a future direction for work for Belinda as a postdoc. She will be able to integrate our Illumina mRNA-seq and the PacBio ISO-seq data on these same samples and follow that up and look for alternative splicing and fusion events and all kinds of things. That's future direction. So I, of course, put this the mRNA changes into Metacore for pathway maps. And we can see a number of signaling that would be found in NAFLD NASH, as we have seen detected phenotypically in the livers. And since M6A is reduced in the Aerochlor 1260 mice, I wondered if writers, readers, and erasers change. So we could look at the differentially expressed gene analysis and look for transcript abundance in integration. And we saw the abundance of FMR, RBM15, and YTHDC2 was actually increased. So Kelly did a Western of YTHDC2, which is a reader. And what we detected was an increase in the Aerochlor 1260 exposed samples. We did this to look also at low versus high NAS scoring livers in these samples. Now, YTHDC2 can be either involved in translation or degradation, so it's going to be context specific. So next steps, what do we need to do? Well, we need to do what's called ECLIP. That's UV cross-linked proteins and nucleic acids, then RNase digest, then IP for YTHDC2. We want to find out what are the transcripts that YTHDC2 is bound to take. When you do phosphatase, you add barcodes to the 3' proteinase K, get rid of the phosphatase. RNA-seq to identify bound transcripts in the control versus the Aeropoor 1260 high NAS livers. So again, this is the direction, but I don't think reviewers will like it because it's too non-mechanistic. It's not, this change is happening and we're going to look at this very thin. It's really, really frustrating to me about reviewers. But I see by some smiles, everybody hears these kinds of things. I then looked at what networks were associated with the Aeropoor 1260 RNA modifications. And here's a pathway. There are the modifications that are indicated. The blue is downregulated. And it's centered in on our Nrf2, which is the mouse gene NFE2. And those of you who are in pharmacology are very familiar with Nrf2 because it is normally inactivated and must stress induced. So then it dissociates from PEEP in the cytoplasm and goes in and upregulates genes that are antioxidants free, involving free radical metabolism and so on to try to get cells to survive. And what we see with our PCB Aeropoor 1260 exposed samples is we see a decrease in Nrf2 in the high mass scoring livers from the Aeropoor 1260 exposed samples. And Josiah had already published work from Matt's lab that in high fat diet Aeropoor exposed samples, they saw a reduction in Nrf2 protein. Now, there were only two changes, I said, that were common, but yet different direction in comparing the low-fat diet Aeropoor 1260 exposed mice versus the high-fat diet Aeropoor 1260 exposed mice. But by doing this experiment, even though the time frame is different, 12 weeks on the high-fat diet versus 36 weeks on the low-fat diet, I wanted to compare just the effect of diet alone on the Epitranscriptome. And my goodness, is it ever different, right? So this is the first identification of diet-dependent differences in the liver Epitranscriptome in mice. So, of course, I ran Metacore and did a network analysis of the epitranscriptomic differences in the livers of the high-fat diet versus the low-fat diet. And it centered in, I'm always interested in what's going on in the nucleus. So I look at it as the center of NFAT TC4. And NFAT TC4 is known to be an activation hub in NASH. We also see PPA5, which is acid phosphatase. It also has a role in NASH. So what happens in NASH? In NASH, you're going to get an activation of NFAT C4. And what activation means is it dissociates from this link RNA called Nron. Because the NFAT gets dephosphorylated, that frees NFAT to enter into the nucleus where it interacts with PPAR alpha and blocks its ability to bind to DNA. So therefore, we're going to get a decrease in fatty acid oxidation. So you're going to start accumulating lipids. And it turns on osteopontin, which is a cytokine that increases inflammation and fibrosis, and this leads to steatohepatitis. So that's pretty interesting. So we looked to find out whether nuclear NFAT was changed in our low-fat diet versus high-fat diet mice. And what we found is that nuclear NFAT is enriched in the livers of the low-fat diet compared to the high-fat diet mice. And we can see that Aeropoor 1260 exposure reduces nuclear NFAT in the low-fat diet-fed mice. So in conclusion from this study, which was just accepted a couple of days ago for publication, and just today I got the little PDF of the accepted paper. Long-term Aeropoor 1260 exposure altered 12 epitranscriptomic marks versus low-fat diet controlled. 18 marks were different between the high-fat diet versus low-fat diet. So diet is regulating M6A deposition, which makes sense. And no, we haven't looked at the SAM levels in the livers or anything like that, or the effect of the gut microbiome. And we confirmed reduced Nrf2 and NFAT TC4 with Aeropoor 1260 exposure. So again, the limitation is that we don't know the location of these post-transcriptional modifications in the global RNA. So future direction. One of the things I didn't really talk about was the fact that in the low-fat diet plus Aeropoor 1260 samples, we saw changes in specific chemical modifications that are unique to selenocysteine tRNA or sec tRNA. And sec-tRNA is the tRNA that deposits selenocysteine into selenoproteins. And changes in sec-tRNA are the major regulators of selenoprotein production. There are 24 selenoproteins in mice, and selenoproteins are critical for liver detoxification of heavy metals and toxins. Toxins like PCB, including Nrf2 in this experiment. So right now I have a CIEHS voucher to test the hypothesis that the observed changes in RNA epitranscriptome in the sec-tRNA are associated with changes in selenoprotein abundance in Nrf2 exposed mouse liver. So my objective is to identify selenoproteins in the liver samples using the expertise of the Omics and Exposure Facility Core for proteome analysis, like Merchant once he gets recovered from his surgery, and to determine if selenoprotein abundance reflects changes in the epitranscriptome of sec-tRNA. I think I've thanked everyone as I went through this talk today. And certainly Belinda, I've been very thankful to have her in my lab. She is just a fantastic student, and it's going to be a great postdoc in working with Julia and others in Eric's group in bioinformatics, and Kelly. And Matt Cave is a very generous collaborator, and I hope to continue to work with him if we can convince NIH to give us some money. And I'll take any questions that you may have.